What is the largest organism on planet Earth? If you're ever on Jeopardy or another quiz show, here's a fun fact to remember. The largest living organism is actually not a blue whale. No, it's a fungus. The honey fungus. Scientific name is Amylaria solipides, and it covers about 6 miles in the Blue Mountains of Oregon. Cool, right? Here's another thing that's kind of cool. The anti-transplant rejection drug, cyclosporin, it's also produced by a fungus, as is one of the most commonly used antibiotics, penicillin. There are some really neat things to know about fungi, and scientists are developing new products that incorporate them. If you're interested in knowing more, do a Google search for Paul Stamets and 6 ways that fungi can save the world. In this video, we want to answer the general question, what is a fungus? Your objectives for this video are to describe the basic characteristics of a fungus, to give some examples of how fungi impact our lives, what kind of roles they play, and then to describe some basic fungal structures such as spores, mycelium, hypha, and fruiting body. Let's start our investigation of the eukaryotes with the fungi. And to help us understand what they are, we'll compare them to organisms that you're already familiar with, bacteria. You'll remember that bacteria are prokaryotes, and fungi are actually eukaryotes. Now, I'm not going to go through the differences between prokaryotes and eukaryotes, but I will point you back to the table that you received back when we were doing the functional anatomy of prokaryotes. Remember, there was a table that compared the prokaryotes to the eukaryotes. You may want to go back to that to get some differences on these different groups of organisms. We also call fungi chemoheterotrophs. Chemoheterotrophs are organisms that secrete enzymes that break down large molecules like complex carbohydrates and proteins and, in some cases, lipids. And they turn them into simple nutrients. For the fungi, it's going to be simple sugars. And those simple sugars can be absorbed through the fungal cell wall. Now, I know you're looking at my table and you're saying, well, it looks like bacteria are chemoheterotrophs as well. And that's true. They are. They do kind of the same thing. It's just that the bacteria metabolize simpler macromolecules than the fungi. Sometimes your bacteria are also what we call autotrophs. So autotrophs are able to synthesize their own sugars. These are usually photosynthetic organisms, or they may be nitrogen fixers. So they're able to take gases out of the air and convert those into usable nutrients. But let's get back to the fungi. I told you that fungi are only chemoheterotrophs, only. So this means that they're going to secrete these enzymes into the environment. The enzymes find a proper substrate, and then they break those large molecules down into smaller ones, which the fungi absorb. If the fungi are getting their nutrients from living organisms, think about, for example, some of the yeasts that live in and on our body, then those are called biotrophs. Bio means life and troph means attracted to. So these are fungal organisms that absorb their nutrients from living organisms. Saprobes are another type of fungus. They absorb nutrients from dead plants or animals. So if you think about those mushrooms that might be growing on an old dead tree stump, they would be saprobes. Or the fungus that seems to bloom from fish that have died in a fish tank and are now floating at the top of the water. Those would be saprobes. Some fungi infect living hosts but kill its cells in order to get nutrients. These organisms are called necrotrophs. Necro, N-E-C-R-O, means death or dead. So these are attracted to the dead. And the cordyceps fungus is an example of a necrotroph. It infects a living organism but slowly kills the organism's cells in order to grow. and reproduce. So I've placed a video in your lecture videos folder about cordyceps fungus. It's called Attack of the Killer Fungi, and I totally think it's worth you watching that video. It's a short one, so take time to watch it. Although fungi may have bright colors and they may have frilly parts that remind us of plants, it's important to know that these are not plants. In fact, fungi are genetically more closely related to animals than they are to plants. And despite their bright colors, fungi don't have photosynthetic pigments. They cannot make their own sugars and nutrients. They have to feed off of other organisms. So again, they are chemoheterotrophs, not autotrophs. Another difference between your bacteria and your fungi is that they're structurally different. So we've already talked about how eukaryotes have membrane-bound organelles like nuclei, endoplasmic reticulum, mitochondria, and Golgi bodies. Well, fungi also have a unique molecule in their plasma membrane called ergosterol. Ergosterol, which is depicted here at the bottom of your slide, is what's called a sterol or a steroid alcohol. It's kind of like cholesterol in our own membranes. It adds fluidity to the fungal membrane and stability as well. Now, why you need to remember ergosterol is as follows. It is only found in fungi. This makes ergosterol and ergosterol synthesis a very popular target for antifungal drugs. Another important difference between your bacteria and your fungi is the cell wall. So your bacteria, remember, have a cell wall that's made up of peptidoglycan. Well, fungal cell walls aren't made of peptidoglycan. They're made up of sugars called glucans and mannans, and they're made up of chitin. You've probably heard of chitin before. It's the same stuff that insect exoskeletons are made of, and shellfish shells are also made up of chitin. Now, because fungi have chitin and these glucans and mannans, and fungi do not have peptidoglycan, that means that antibiotics that target peptidoglycan synthesis, like penicillin, are not effective against fungi. So this will be important later because some fungal infections look like bacterial infections. But if you try to treat those fungal infections with the same antibiotics that you would use for a bacterium, it's not going to work because fungi have different molecules in them, like this chitin. Now, catabolism. Catabolism refers to the ability of organisms to break down large molecules into small ones. And catabolism is one half of the equation for metabolism. It turns out that fungi are less diverse than the bacteria are with respect to metabolism. Remember, in addition to the chemotroph versus autotroph, you also have several different types of metabolism and thus oxygen requirements for the bacteria. Fungi, on the other hand, basically do only aerobic cellular respiration or a very specific type of fermentation. So they're typically obligate aerobes or facultative anaerobes only. You won't find obligate anaerobes or microaerophiles or aerotolerants in the fungal kingdom. So that's another difference. With respect to reproduction, your prokaryotes do primarily binary fission. Fungi primarily do mitosis and meiosis. And then finally, both your bacteria and your fungi make spores, but the function of those spores is different. Remember, when we talked about bacteria, we said they make these fungal cells called endospores. And we said that endospores are primarily used for survival. So one cell makes an endospore when, say, essential nutrients are missing or when the environment does not support growth. And one cell emerges from that endospore. Fungi, on the other hand, make spores to reproduce, and they have sexual spores and they have asexual spores. One sac of spores from a mold can produce 50,000 new fungi. That's mind-blowing, isn't it? Why do they produce so many spores to make so many new fungi? Well, they rely on the air to carry the spores to new locations where the fungi can germinate. And if a spore lands on a surface like a desktop, it's not going to make a new fungus. So these organisms need to make lots of spores to give themselves the best chance at reproduction. So fungal spores are used for reproduction, not just survival. The differences between bacteria and fungi extend beyond what I mentioned in the last slide. Some examples are fungi have adapted to live in environments that won't support the growth of bacteria. For example, fungi like to live at a pH of around 5, which is much more acidic than the pH of 6.5 to 7.5 that we see with bacteria. Your fungi are also capable at existing on lower nitrogen levels than the bacteria. Fungi are also more resistant to osmotic pressure. So this means that they can grow in hypertonic environments that inhibit the growth of bacteria. I think about, for example, how jams, jellies, and preserves occasionally mold, but it's unusual for them to go bad because of bacterial activity. And these, by the way, are properly prepared jams, jellies, and preserves. Your fungi also can grow on surfaces with low moisture. I think about, for example, shoes that may be stuck at the back of a closet that begin to mold after a year or two of just sitting there, or old clothes that mold because they've been left in the basement. Okay. Another difference is that your fungi can catabolize more complex carbohydrates than your bacteria do. So this means that there are some environments where fungi will grow that bacteria just can't because they don't have the enzymes to break down the carbs in the environment. Now, when we talk about fungi, we're talking about any of about 144,000 known species of organisms that belong to the kingdom fungi. This includes yeasts, smuts, rusts, mildews, molds, and mushrooms. I've listed some of their benefits and detriments on this slide, but I'll only highlight a few of them. One benefit of fungi is that they are the major eukaryotic decomposers in our environment. They break down dead plant and animal matter and release nutrients that can be absorbed by other organisms like plants and bacteria to support their growth. This is a form of nutrient recycling that's essential to the circle of life. Mycorrhizae are particularly important in this. These fungi are in symbiosis with plant roots, and they help the plant roots absorb water and elements like nitrogen, sulfur, phosphorus, potassium, as well as macronutrients from soil. This benefits plant growth. In return, the mycorrhizae absorb sugars and other nutrients from the plant. If you look in the gardening section at your local Home Depot or Lowe's, you'll probably see products containing these fungi for sale. The idea is that they'll help your garden plants grow. Yeasts are master fermenters. They take sugar and turn it into alcohol, ethanol specifically, and carbon dioxide, which is why they're important ingredients in bread and alcohol production. Or they turn sugar into lactic acid, which is why they're important in making sourdough bread. In addition to penicillin, fungi produce medications like Pravachol and Mevestatin, which are cholesterol-lowering drugs, cyclosporin A, which inhibits organ transplant rejection, and lentinan, a drug that may enhance immune system function to reduce the growth of certain tumors for cancer patients. Now, a small number of fungi are responsible for causing disease in humans, animals, and to a larger extent, plants. It's estimated that fungal diseases are responsible for about 1.6 million deaths worldwide annually, and about 1 million people, not 1 million, I'm sorry, 1 billion people suffer from severe fungal disease every year. Fungal diseases threaten bats. You might remember that white nose disease in bats that was in the news a few years ago. They threaten amphibians and reptiles as well. In addition, it's estimated that fungal pathogens destroy 1/3 of all food crops worldwide. If you garden, you're probably familiar with the heartbreak of finding late blight on your tomato or potato plants after nurturing them for several weeks. Fungal diseases in plants cost about $60 billion in lost revenue annually. Now let's switch gears and talk about fungal structures. Your fungi are either unicellular, meaning the fungus is one cell, or they are multicellular, meaning that the fungus is composed of many cells. Your multicellular fungus makes two types of structures that you're especially responsible for knowing: the mycelium and the hypha. Unicellular fungi are usually called yeasts. Let's talk about the multicellular fungi first. Hyphae are microscopic, hair-like tubes that make up your multicellular fungi. They're colorless, thin, branching threads. And they tend to run throughout the material or the substrate that the fungus is growing on. These types of hyphae are called vegetative hyphae, and their primary job is nutrient acquisition. So these are the hyphae that secrete enzymes into the environment to break down complex carbohydrates like lignin. And then these are the same hyphae that absorb the simple sugars that result. You can see some examples of hyphae in this picture here. The threads that you see are little bundles of hyphae. Another type of hypha that a fungus will make is called an aerial hypha. Okay, aerial hypha stick up from a substrate and they have a couple of functions. They act like breathing tubes and absorb oxygen when it's present to support the metabolism in fungal cells. Other aerial hypha contain reproductive structures that make spores. When you see structures like the ones in this picture or in this picture, you're looking at bundles or clumps of hypha. When these hypha grow together, they form a roughly circular mat. This is just a portion of one called a mycelium. Mycelia are usually hidden from our view because they're embedded in the surface that the fungus is growing on. But you might have seen a mycelium or two on molding bread. So what about mushrooms, Professor? Aren't they fungi? Yes, 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 they are. They are the fruiting bodies of a fungal mycelium. So these structures, the fruiting body, is specifically designed by nature to protect spores and to lift them up into the air where they might be carried on the wind to new locations. So what you're looking at here in this mushroom is bundles of hyphae that forms the stalk and the sporocarp here. And then under the sporocarp, you have gills. And in these gills, you have these little pedestal-like structures that hold the spores. At the appropriate time, the gills open up and the spores either fall out onto the ground or they fall out and get carried on air currents to other locations where they may germinate to form a new fungus. Have you ever seen one of these? This is called a fairy ring, And when I was a kid, people used to say that fairies would dance inside the ring at night, or they might sit on the mushrooms that form the ring. Now I know that this is not a fairy, not a meeting spot for fairies at all. Rather, what's happening is a spore from a fungus has landed in the middle of the ring, and then as hyphae begin to grow out in all directions from that spore, you get a circular mycelium. These fruiting bodies, aka mushrooms, form at the very outside edge of the mycelium to produce spores and keep the mycelium growing. So that's where the fairy rings come from. Before we move away from fungal structure, I do want you to know that there are two types of hypha. So I talked about the functional types, we talked about the aerial versus the vegetative, but structurally there are two types of hypha as well. The first type is depicted here, and this is called a septate hypha. Okay, septate. So what's going on here is that the hypha is made up of multiple cells. And in a septate hypha, the cells are separated by a partition called a septum. And what's interesting about this is that there's a pore that goes through each septum, connecting the cytoplasms of all of the cells together. So this allows for the exchange of chemical signals through the hypha, as well as nutrients. A second type of hypha that we see looks like this. It's called cynocytic. Okay, cynocytic. And basically in this type of hypha, we have multiple nuclei as well, but there's no, multiple cells as well, but there's no septum to separate the cells. They're just like one long continuous structure. Okay, so that's cynocytic. And again, if you look at this image, you don't really see any partitions at all. So as I've already mentioned, in addition to your multicellular fungi, you also have unicellular fungi, and they're called yeasts. Yeasts are divided into two categories based upon how they reproduce. So you have fission yeasts, which basically do a process that's very similar to binary fission. The yeast cell grows and then it develops a partition in the middle that splits the cell into two. But you also have budding yeasts. Saccharomyces cerevisiae, which is responsible for making bread and beer, it's a budding yeast. So here, a little bleb or bubble forms on the surface of the yeast. The yeast cell fills that with cytoplasm and eventually a copy of its DNA. And then that little bleb grows and grows and grows, forming what's called a bud. Eventually, enzymes break the cell wall between the bud and the parent cell, and the bud breaks off, leaving behind a little scar called a bud scar. So we can see how many times a cell has reproduced because it'll have a little scar for each one, kind of like a belly button. In any case, these are your budding yeasts. Some yeasts are what we call dimorphic, and they tend to be pathogens. Dimorphic fungi go between being yeasts and being like molds, making hypha. So at 37 degrees Celsius, you get the yeast-like form, which you see over here. All of these little circles are individual fungal cells. They look kind of like the buds that we see in the previous picture up here. But as we lower the temperature that this yeast is growing in, we start to see the development of hypha-like structures. So the fungus switches from a yeast-like growth profile to one that's more like molds with hypha and mycelia. Now let's talk about fungal reproduction. Fungi, like many eukaryotes, are capable of both sexual and asexual reproduction. In asexual reproduction, one fungus produces spores after completing mitosis. And usually fungi use this type of reproduction when there are lots of nutrients around and the environment is favorable for growth. Why? Well, if you're a fungus, this is the fastest way of reproducing yourself. You only need one set of chromosomes and you do mitosis over and over and over again, producing lots and lots of spores. Remember that 50,000 number that I told you a while ago? That's the result of this asexual reproduction. Once all of those thousands of spores are produced, they can be released to the wind. And then the wind will carry the spores to other locations where, if the spores land on the right surface, they'll begin to grow new fungus. Okay. Asexual spores are the ones that are usually formed by human pathogens, and at one time they were commonly used for clinical identifications of fungal diseases. Sexual spores are formed when two different fungi, their nuclei, fuse together. So you have two hypha from two different fungi. One hypha penetrates the other hypha and donates a nucleus to that hypha. The nuclei from both fungi fuse together, mix, and then you get meiosis, which partitions out the chromosomes again. Okay, and then that makes a sexual spore. Sexual spores tend to be made when conditions around a fungus are not suitable for growth. For example, maybe there are few nutrients, or maybe the environment is extra dry for a prolonged period of time. Sexual spores are made for survival. They are resting spores. These spores sometimes feature thicker cell walls, and they're usually stored in fruiting bodies, aka mushrooms. Fruiting bodies are structures that can be dried, and they're able to resist damage from the environment, so that also protects the spores and helps them to last longer. Spores can be formed on hypha, in structures called sporangia. They can be formed in sacs called asci, and of course they can be formed on fruiting bodies. So now let's take a look at some of these asexual spores and how they're produced. The first type of asexual spore is called a conidia. So all of these little spheres that you see here, these are conidia, and they are formed on a special aerial hypha called a conidia 4. For your conidia, they're actually formed as chains. You can kind of see if you look at this particular structure, you can kind of see that the conidia are forming a chain-like structure. Okay, and these chains are at the end of a conidiophore. Examples of fungi that reproduce this way include Aspergillus and Cryptococcus. Another type of spore that gets produced asexually is called an arthrospore. So here you have lots and lots of hyphae. They're kind of bundled together, and these hyphae are septate. At kind of the end of the hypha, the cell wall of one of the segments thickens, producing a slightly thickened cell called an arthrospore. And these thickened cells can break off or fragment, which means that they can go on to produce new fungi. So these are an example of conidia, but they're formed by fragmentation of septate hyphae. Okay. Examples of fungi that reproduce this way include trichophyton, which is associated with athlete's foot, and coccidioides. Pseudohyphae are formed by yeasts like Candida or fungi like Cryptococcus. Here you have buds that are produced. So remember that budding reproduction that we talked about? You get a bud, but the bud doesn't actually break off. It fails to detach itself from the parent. And so you get a short chain of connected buds. Okay, and then because it's similar to a hypha, but it's not really a hypha, it's called a pseudohypha. Blastoconidia are another type of reproductive cell or spore. These are the buds that actually come off of a parent cell. Chlamydioconidium are spores that are formed by the rounding and enlargement of a particular part of a hypha, like this. So we see this sometimes with Candida yeast. This is a cool picture because you can see pseudohyphae down here as well as this chlamydoconidium up here. Sporangiospores are small spores. So you can see how tiny these guys are. They're smaller than the others that we've looked at. And they're formed in a sac. So they're actually formed inside of the fungus on the end of an aerial hypha called a sporangiophore. These spores get released when the fungus or the mold, in this case, is brushed and the sac ruptures. Bread mold reproduces this way. This is when you look at a bread mold and you see those little tiny black balls, this is what you're looking at. So prior to about the 1990s, maybe even the year 2000, most clinical identifications of fungal mycoses, that is your fungal diseases, required the removal of a specimen from a patient. And the type of specimen depended upon what kind of disease the patient presented with. So it could have included like a skin or a nail scraping with a special tool, or a sputum culture where thick mucus from the lung was expectorated after a cough. Maybe it was a blood specimen or even a urine specimen. Then that specimen would have been smeared onto a slide and stained, or it would have been sent to a lab where the fungus would be cultured, and then technician would remove a little bit of that and put it on a slide and stain it. And then they'd look at spore forming structures like these, or look for fungal cells like these under the microscope and try to identify what fungal disease, what mycosis a patient had. Nowadays, identification of fungal spores using a microscope is on the decline as a technique. There's a lot of factors that play into this. One is that it seems like more people are affected by fungal diseases, and at the same time, there are fewer professionals that specialize in fungal spore identification. Add to that, we have better tools like PCR and serology, where the fungal spores are reacted with antibodies. All of this gives a more specific ID than microscopic examination might give. So now we've reached the end of this video. I've given you the basic characteristics of a fungus in a table at the very beginning of this slide presentation. You'll want to remember that your fungi are eukaryotes and they're chemoheterotrophs. Their way of getting nutrients is to secrete enzymes into the environment and absorb the breakdown products, the simple sugars that are released. Then they use that to drive their growth. You'll want to remember that your fungal cells have some unique structural characteristics. They have ergosterol and they have chitin, both of which are popular targets for antifungal drugs because these two molecules are either non-existent in other organisms, like we see with ergosterol, or it's rare. So remember, chitin is only found in like a handful of animals. I gave you some examples of how fungi impact our lives. I listed some drugs that they produce. I listed their role as decomposers. I also talked about crop damage and how they're responsible for fungal diseases. Finally, we talked about fungal structures. We looked at spores and talked about how they're used for reproduction. We talked a little bit about how a mycelium is basically a mat of fungi, and it has a circular form because typically when a spore lands on a suitable surface or substrate that provides nutrients, hyphae begin to grow in all directions from the point where that spore landed, and this gives us this circular mycelium structure. We learned about the different types of hyphae, both in terms of function as well as in structure, and we talked about mushrooms and said that they were fruiting bodies meant to protect spores in unfavorable situations, but as you and I know, they're also pretty tasty, or can be. So in the next video, we're going to start our investigation of fungal diseases. We'll talk about how fungal diseases, aka mycoses, are characterized, and then we'll actually look at some common fungal diseases. If you have any questions about this lecture or any other, please don't hesitate to reach out to me by text, email, or phone, although that's probably the least effective way of reaching me, and I'm happy to talk about this with you. Thanks for watching.